Good morning, and thanks to the organizers for the kind invitation to speak. Here are my disclosures. So I'm here to talk about UTI, which we all know about, and it's almost so common that we don't sometimes stop to think about how complicated it is. So recent figures suggest 400 million cases per annum. 400 million, that's a lot of people. And as we've been talking about AMR, the fourth top reason that people die from infections that are associated with or attributed to UTI, attributed to AMR, so 250,000 people dying from UTIs associated with AMR. And for me, one of the most interesting things that I'm a basic scientist, and in our laboratory, we're really interested in recurrence. 20 to 30% of UTIs will recur despite treatment. So why is that? It's quite mysterious. And of course, it largely affects women, and 1 in 2 women will get one in their lifetime. And before I talk about the science, I just want to say one thing. It's a little bit of a feminist rant. I hope you'll forgive me. It's been nearly a century since Fleming discovered penicillin, and we still don't have a better first-line therapy for UTI. And this affects 400 million people, and 20 to 30% recur. So it's not a very good therapy. It's been nearly 70 years since Cass sort of formulated the diagnostic criteria of the midstream urine culture, which is really not that dissimilar for what Fleming was doing a century ago with his Petri dishes. We still don't have, I would argue, better diagnostics. Why is this? And I think it's obvious that it's because it's primarily a disease of women. Many studies have shown that we have a very high cultural tolerance for women suffering. A quote on there, I was told that my pain was made up for 7 years. You hear things like this all the time. And research funding is disproportionately showered on diseases that are not primarily affecting women. So I'm not going to talk any more about this, but I just wanted to put this in the proper context because it does make me a little bit angry. Now on to the science. So UTI is deceptively simple, but it's actually quite complicated because it has a really interesting life cycle. The bugs start out in the gut, and they make that journey from the back passage into the urethra, which is open to the environment. The bugs can colonize the bladder, causing cystitis, occasionally ascending to the kidneys to cause more serious infections, kidney infections, and potentially sepsis. And we think about UTIs maybe being a one-size-fits-all thing, but there are actually many dozens of microorganisms that can cause UTI. We think about E. coli, uropathogenic E. coli, or UPEC as being the most common, but there's lots of other bugs, especially when you get into more vulnerable or complicated cases or the elderly. So we know hardly anything about UTI caused by UPEC. There's about 24 labs in the world that study UTI, believe it or not. And amongst those bugs that are not E. coli, we know even less. So it's a very complicated life cycle, very complicated different kinds of bugs, lots of diversity, and hardly any research at all. So in our lab, we're trying to make it a small little inroad into this problem. What we like to do is to look at patient samples of bacteria, but we take them into the laboratory and we study them in human-based model systems, and I'll talk more about that in a minute. So we can do really highly controlled experiments, the experiments that we can't do on human beings because it's not ethical. And then we're trying to make new therapies. And we study a lot of different things in the lab, and I don't have time to talk a lot about it, but what I want to focus on is on the things that are contributing to the treatment resistance and to the diagnostic problems that we have. So just before I talk about the science, to orient you on the battleground that I'm talking about here, this is the bladder. If you take a cross-section of the bladder wall, you see this beautiful stratified epithelium, so layers of cells, and the urine is on the top here in the black area, and that's where the bacteria come in, and then the urine is flowing from the kidneys out, and the basal side is the muscle side. So this is what we're talking about, the urothelium. And this trick is not working so well. Come on. Okay, so 20 years ago, this is the sum total of what people thought about UTI. They thought, okay, the bugs come in, they dock on the surface, they cause a bit of inflammation, the neutrophil, the cavalry is called, and the neutrophils arrive and hoover them up, and then, of course, you urinate out the bugs as well. This is the sum total, and this made it really weird. Why is it that we can't treat this very simple infection? And, of course, the answer is that it isn't that simple after all. 20 years later, with some research, we now know it's very complicated. Don't worry, I'm not going to take you through this whole slide, but I want you to visually appreciate the difference between this and this. There's a lot more going on than we originally thought. I'm just going to draw your attention to a few things. We know that the bacteria that cause UTI can actually burrow into your bladder wall. take residence inside the cells of your body and hide there and form these massive colonies called pods, which can burst open like alien later and cause a reinfection. We know that the bladder, the healthy bladder, is full of bacteria. So we have a microbiota in the urine, which is, of course, a little bit of a complication when it comes to diagnosis. And we've got an adaptive response, and we've got biofilms and all sorts of things. So I'm going to focus on just a few of these complications to illustrate my point. The first thing I want to talk about is the intracellular pods, these sort of horrific things that are hiding in people's bladders. Okay, so it's a very compelling idea, and it was first noticed in mice. If you give mice a UTI, you almost always get these pods forming. So this is an example. These green things are bacteria. They're inside the cell of the mouse bladder. The colony here is so big that it's bigger than the cell nucleus, so it's a massive colony. And you can see these all the time in mice, stereotypically, reproducibly. In human cell lines, so if you take cancer cell lines and you try to infect them, you can see these intracellular bacterial communities. It's quite easy, but cell lines are not people. In actual human beings, the data are very scarce. So we and others have shown that if you collect cells shed in the urine of patients, you can find, see those blue things, you can find bacteria hiding within those cells. And this is the one study I know of that was actually a biopsy study of infected women. And you can see, this is a cross-section of the bladder. You can see those little green dots. Those are the bacteria inside the bladder wall, hiding. And they're not in the urine, they're in the bladder wall. So that's really compelling, but it's only one paper. How common is this? So we wanted to build a better model to see if we could study this in the lab. The reason why is because mice are great, and people do lots of experiments on them, but for urinary tract infection, it's not a very good model. The urothelium on the right here is a lot different than the human urothelium. It's very thin. Urine is highly concentrated in mice, and mice don't do the thing that we do, which is go down to the pub, I'm not speaking for myself, but drinking 4 pints of lager and storing all that urine and distending your bladder. I hope no one needs the loo. Whenever I give this talk, people are rushing to the loo. Sorry about that. So that we have storage, a distension and a storage, whereas mice just tend to spritz their urine territorially and they don't store. So there's physiological differences, urodynamic differences, and even the markers on the surface are different. So it's a very different model. And most crucially, it's really hard to give a mouse a UTI. They don't get them naturally. You have to put a tiny little catheter up the urethra, sort of a venous capillary, and put billions of bacteria in their bladder, and then they get an infection, but they don't naturally get it. So it's not a good model. So we wanted to build our own model. We wanted to grow human mini bladders in the lab. And to do this, we used a little structure here. It's called a transwell. We seed stem cells onto it and develop a whole little bladder urothelium onto this thing. It's about the size of a 5p coin. And if you look at you basically have, it's bathed in growth medium, and you can put 100% bona fide human urine on top, and you can do experiments on top of this mini bladder. It's a really nice model. It's fully differentiated. It's got the thickness of a human urothelium. It even produces a gag layer, so it's got this mucus on it. It produces chemicals in response to infection, defense chemicals, and it's urine tolerant for weeks. So you can do lots of experiments in these mini bladders. And there's just some pretty pictures of it. And this is an example of an infected mini bladder. So the green things are bacteria that we put on top, and they're in the process of docking and invading this bladder. Okay, so what did we find? When we did our experiments, we were kind of surprised, but very delighted to see that we got massive invasion of our bladder model. So just like what is seen in mice, you can see that the bacteria go inside the bladder wall. And just to orient you, the urine side is on the top, and this is a cross-section of the urothelium. So if there was a detrusor muscle, it would be down here. And you can see the green things are bacteria that are only on the outside. And the big blue, I've circumscribed them with a white dotted line. These are massive intracellular colonies of E. coli. And if you get your eye in, you can see they're all over the place. Lots and lots of bacteria inside the bladder wall. And then these dense blue bodies are the nucleus, or the human DNA. So yes, we're getting lots of invasion, and we're getting it with multiple different kinds of bacteria, not just E. coli. So this seems to be a thing. Invasion of human cells seems to be a thing, which is interesting. It has implications. So the first implication is if all of your bacteria are in the bladder wall, and you give a urine specimen, there may not be many bacteria free in the urine. So you might get a negative MSU, even though you're actually riddled with bacteria. So that's the first thing to keep in mind. The second thing is that antibiotics really struggle to penetrate the bladder wall, which is armor-plated. It's designed to resist the influx of urine. Urine is toxic. You don't want that in your tissues. So it's really hard for antibiotics to get in there and accumulate and kill these things. So this could be another explanation for why it's hard to treat UTI, all because of the biology of the bugs. Okay, now let's talk about biofilms. We think about biofilms a lot in terms of catheter-induced UTI. So there's always manky catheters, and there's lots of bugs, and they're really dangerous. But people don't think about biofilms in a sort of a non-catheterized situation. But if you think about it, biofilms, or communities of bacteria that like to group together and form a slimy layer, biofilms are very common in nature, very common in chronic infections, especially when there's fluid and fluid flow. So the bladder, there's lots of fluid flow there. So it's thought that there should be biofilms just free in the bladder like this on the surface. And when we looked in our model, here again you can see in gray is the surface of the model. We're looking at top-down view now, bird's eye view. The orange things are Pseudomonas in this case, or Proteus on the right, and that acid yellow color is describing the sticky matrix that they produce when they make a biofilm. So both of these prolific biofilm formers are making biofilms on the surface of our bladder model. And I want to stress that these biofilms are really sticky. It's really hard to wash them off. So if you were urinating, you wouldn't necessarily get rid of any of these biofilms. So again, we have the implication, you may not get these bacteria showing up in your midstream urine culture. And again, antibiotics really struggle to treat biofilms. Not only do they struggle to get through that acid green sticky stuff, but if they do get inside, the bacteria tend to be dormant and they're not presenting the target for antibiotics, which is usually something like cell division or some other active form of metabolism. So biofilms are really, really hard to treat. Again, because of the behavior of the bugs, we have an explanation, perhaps both why they're hard to diagnose and hard to treat. And the third thing I want to talk about is the urobiome, or the microbiome of the urine, which is really a complicating factor if you want to use more sensitive techniques like genomics to diagnose your UTI, because there are other bugs up there. And we did a study of, a few years back now, of patients who had lower urinary tract symptoms that felt like a UTI, and they had white blood cells in their urine. So there was evidence that there was infection, but they tended to be testing negative on the midstream urine culture, and certainly on the dipstick, which isn't very sensitive at all. And when we looked at them, patients on the left, controls on the right, we found lots of bugs in the patients and in the controls. So the controls, people like you and me who don't have an infection, would have Enterococcus, they would have E. coli, they would have Klebsiella. So there are actually uropathogenic species in healthy urine, but that doesn't help with diagnosis. Other key findings from this study, routine MSG was actually failing to detect a lot of uropathogens in these patients with symptoms. We could see those in both patients and controls, so it wasn't very discriminatory. The MSG was not able to discriminate between symptomatic and non-symptomatic people, which is a bit worrying because it's not very sensitive. Different sets of bacteria were detected. If we use midstream urine culture, we got one set. If we use sequencing, we got a different set of bugs. So what's the right answer? The genomics or the midstream urine culture? Pass, I have no idea. So MSU might be unsuitable for excluding UTI. If you've got a case where you have maybe a low-level infection or a chronic infection, if you've got an acute UTI, you don't really need a test, do you? It's obvious from the urine. But if you've got a sort of low-grade or chronic recurrent UTI, it might be more difficult. And one final thing I want to point out from the study is that mixed growths in this study were clinically significant. There is no reason to think that UTI cannot be caused by more than one bacteria. And in fact, there are bacteria that are known to work together, like E. coli and Enterococcus. They like to travel together, they work together. So when you get a mixed growth report, it's often discarded as contamination, but sometimes those mixed growths are significant, so that's worth keeping in mind as well. Okay, and just to mention something really interesting that's happening in the Royal Free Hospital, where my unit is based. So we look at a lot of renal transplant recipients. These people are immunosuppressed. They get lots of UTIs, and they're really life-threatening and graft-threatening. So they take a lot of care with treating these patients. And we keep seeing these sorts of things. So a person comes in with an E. coli. You do an MIC in a midstream urine. You determine that the bug should be susceptible to antibiotics. Less than a month later, the same patient is back with the same bug, and the bug is still sensitive on paper to that antibiotic, but the antibiotic, the correct antibiotic, did not work in that patient. So relapse, in other words. So that's really interesting. And we thought maybe we could take a look at this in our model. We can use this as an infection model. So we can take our mini bladders, we can infect them with, for example, E. coli, then we can treat them with a drug it's sensitive to. We do an MIC, and we determine that nitrofurantoin is a good choice. So we treat them with nitro, and then 24 hours later, we see what happened. Did it kill the bacteria or not? In the MIC, it worked really well. Does it work in the mini bladder? The answer is no. So whether you look at the free-floating bacteria in the urine or you look at the bacteria that are associated with the bladder wall, it takes up to 20 times the MIC to even start to see an effect. So the higher the bar, the more growth. What you want to see is that bar go down to 0, total killing. But at the MIC, 5 times the MIC, 10 times the MIC, nothing. And then at 20 times, a bit of a dip, but we still have nowhere near eradicating this bug. You could explain that, with some of the things that we've already talked about today. So in the bladder wall, it could be the biofilms, it could be the intracellular bacteria. That could be the reason why the treatment is not working. But what's happening in the urine? So there's no excuse. There's no biofilms in there. There's no intracellular bacteria. It's known, actually, that there can be genetic reasons. So a bug that's otherwise sensitive to an antibiotic can temporarily switch on genes that make them resist for a while. They're not really resistant. They're not AMR, but they're sort of resilient is the word we use. And we've been seeing some evidence that this might be happening at the RNA level. So bugs, even though they're genetically sensitive, and the test says use this drug, may not be killable in real life. Okay, and it could be that different patients have different combinations of reasons why they fail treatment, and there may be other reasons as well. So it's all very complicated. So just to say, I think it's important to understand then, how long should we treat? We were talking about underdosing before, and I think there could be a case that we might be underdosing UTI. 3 days of antibiotics, I'm not convinced, is enough to get rid of the bugs, considering what we're actually seeing in the bladder situation. And there's a really interesting trial. I'm not involved in this trial. It's coming out of Oxford. It's called Durations. They simply want to determine how long do you have to treat a UTI so that it doesn't come back, which is a great experiment. I've been hoping somebody would do this. Unfortunately, they're only looking for 42 days follow-up, and as we know, a recurrent UTI is defined as having 2 every 6 months or 3 in a year. So I'm not sure 42 days is long enough, but it's a start. Because I want to know, what is the initial treatment length you need to clobber it so it never comes back? Because if you only treat for 3 days, it's possible that you'll just get relapses, and instead of just killing it off once and for all, with maybe a longer course, you end up with a person who gets it again and again and again for years. And if you consider using maybe a 7 or 14-day course initially and never having to use it again versus undertreating and then never getting rid of it, I think it's clear from both the AMR point of view and the resource point of view what's the better strategy. So that's a really interesting trial. I'm looking forward to the results of that. I always like to end my talks on a note of hope, because it's a bit of a bummer, my talk. Right? We basically have this disease of 400 million people, mostly women, really hard to treat. There aren't any good diagnostics or treatments, I would argue, at the moment. So there's some interesting things coming down the pipeline. You probably all know about diagnose. A recent study showed it doesn't work very well, but there are companies that are riffing off the design of diagnose and trying to come up with bug blockers that will stop E. coli and Klebsiella and gram-negatives in the gut from actually binding in the gut and maybe getting rid of them at source. So those look really promising. I think probiotics is a totally exciting area, inspired by sort of fecal microbiota transplant. Maybe these kind of transplants would help recurrent UTI sufferers as well. Use the good bugs to fight out and outcompete the bad bugs without using any drugs at all. That would be brilliant. I think phage therapy is making a comeback. This is a very ancient technique from the former Soviet Socialist Republic. It's now having a sort of a renaissance of interest again. I mean, there are a lot of challenges with phage therapy, but it's a great idea, right? Alien versus predator, right? Use the bugs, the viruses to fight the bugs. And finally, I think vaccines are exciting. It was only recently appreciated that UTI has an adaptive immune response. And I think Euroimmune looks really exciting. There was just a really interesting announcement at a recent conference, like a 9-year follow-up, 50% of recurrent UTI sufferers seem to be cured. So watch this space. I think it's really exciting. Just to summarize, human cell models are useful, I hope I've convinced you, for studying biology and treatment and may advance our knowledge. There are biological reasons why UTI is hard to diagnose, and actually technical reasons. I didn't have time to talk about it, but the standard tests are, I don't think, fit for purpose at the moment. And there are biological reasons why treatment fails as well. one size may not fit all. Patient A might need a different treatment than patient B, but that's not what they get at the moment. So we need more awareness, we need more research funding, we need better diagnostics and treatments for UTI, especially non-routine UTI. That's it, this is just my lab, and acknowledging my funders and my collaborators. Thank you.